Hi, I'm Jay Phelan. Let's talk about some biology. We're going to talk today about another of the variants of what's referred to as modifications to Mendelian ratios or extensions to Mendel. And the topic today is going to be called epistasis. And we'll start in by making the point that, as I've said before, that Mendel looked at many traits that were coded for by a single gene that had two separate alleles. One was completely dominant to the other, and there was no environmental effects at all. And that helped us figure out genetics, the transmission of information from parents to offspring. But we're trying to build a more complex model of how genotypes get turned into phenotypes. And to do that, we have to look at many different situations, such as when there are multiple genes that can influence a single trait. That's what we're going to look at today. Let's say that you're looking at something like the flower that's called the blue-eyed Mary. And typically, the flower petals are blue. And then let's say suddenly you see a bunch of white ones. Some plants arise in your population. They've got white ones. So you're a good geneticist. What are you going to do? You're going to cross them. You're going to say, what's going on here? How is color determined in the blue-eyed Mary? So you cross the blue-flowered plants, the white-flowered plants, and you find out that the next generation, we'll call it the F1 generation, they all have blue flowers. Okay, we're doing this because we want to figure out what is the mode of inheritance. You cross all of those F1 individuals, these are blue individuals, and now you get, let's say if you look at 134 plants, you got 101 blue, 33 white. Those F2, that generation, we'd say, it looks like there's about 3 times as many of the blue as the white. We always want to assume that the simplest explanation is most likely to be true. So in this case, our interpretation would be, all right, maybe there's a single gene that codes for color, and white is a recessive trait. So there's a recessive albino trait. If you're homozygous recessive, then you're white. That would mean that our F1 cross here was a cross of individuals that were heterozygous, hence the ratio that we got for F2. But let's say someone else in some other population comes across some purple flowers in this species, blue-eyed Mary, and they're trying to figure out, all right, well, why do we get purple sometimes? And they do what any geneticist would do. They cross them. They cross the blue and the purple, and when you cross those, they find the F1s, the offspring, all have blue flowers. It's kind of the same thing going on again. So you take those blue-flowered plants, cross those with each other, and what do you get? They get a similar kind of thing. They got 192 blue, 63 purple. We look at that, we're like, it looks like the blue are about 3 times as common as the purple. So in that F2, we're thinking, all right, 3 to 1. I think it's probably the same thing going on. It looks like a single gene that codes for a recessive albino trait that is the purple. Simple explanation. But in this species, you'd say, all right, well, what's going on with the purple and the white? Is this some kind of complementation? Same mutation, other gene is responsible for that. So you do what any geneticist would do. You cross the purple ones and the white ones. See what we get. Well, if we do this, here's what we find. We find that the offspring are all blue. So we go back to that most common trait that we see. We're going to cross the F1 offspring. When we cross those, now we get a strange thing that we haven't seen before. We didn't see with complementation. We don't see with a standard Mendelian cross. We find that when we cross those F1s, the blue flowers, we get a ratio of about 9 blue to 3 purple to 4 white. We want to know what's going on. Well, given that these ratios, as you can see, are some modification of 9 to 3 to 3 to 1, we think, it's not really obvious that, but we say there's probably 2 genes involved. Because remember, the 4 by 4 Punnett square, when you cross individuals that are heterozygous for 2 different genes with another individual heterozygous for 2 different genes, and you look at all the possible genotypes you get, we find that there are a ratio of phenotypes of 9 showing the dominant for both, 3 showing the dominant for one, recessive for the other, 3 showing the reverse of that, and 1 out of the 16 showing the recessive for both. So we see this ratio and we think something's going on here that could be explained by a situation where we've done a cross that was a cross of individuals heterozygous for each of 2 alleles. So standard situation, you see this a lot. You cross double heterozygotes, get 9 to 3 to 3 to 1. I use A and B. They can stand for anything. The line refers to an allele that could be a dominant or allele. Doesn't matter. I have a big A and then a line because once you have one big A, if that's dominant, doesn't matter if the other is big A or little a, it's going to show the dominant phenotype for A. Same thing goes for B. So we have these, and if we try to now reconcile our cross of the purple flowers and the white flowers with a 2-gene situation, we could say this. We could say, all right, the 9 out of the 16, clearly that seems to be a phenotype that reflects a genotype where you've got at least one dominant allele for both of the genes. So our first case here, we think when you end up with one of those genotypes, you're going to be blue. Now for the others, the 3 and the 4, well, 3 purple. Let's just say the next one. All right, you're homozygous recessive for the second gene, but for the first one, you've got at least one dominant allele. That leaves 4 remaining, and we see in this case, all right, the 4 that are remaining are the ones, it doesn't matter what you have for B. If you have little a, then you're going to end up white. And that's the key here to epistasis. It's looking at this situation and saying, every time there's homozygous recessive for one of these genes, it doesn't matter what you have for the other one, you're just going to be white. So a way of articulating this would be to say that the presence of the homozygous recessive genotype for this other gene, this epistatic gene, masks the effect of the other one. It no longer matters what you have for the other one because this one is going to cover it up. So the ratio 9 to 3 to 4 when you do a double heterozygote cross, that's called recessive epistasis. And just articulating what I said before, that when one of the genes is homozygous recessive, cancels out the other gene regardless of which alleles are present for that other gene. Now, what if you had a situation where instead of little a blocking out whatever happened at the other gene, what if for that first gene it was a dominant situation? As long as you had one big A, you were going to knock out what went on with the other one. So we're going to look at that. It's called dominant epistasis, obviously. So we could take an individual that is true breeding dark red, flowers, say, cross with one that is true breeding and white, and they are showing this phenotype because of what they have for one or the other gene. When we cross these, they'd become doubly heterozygous. They're all white. We cross those F1, so we're doing a double heterozygote cross like we just looked at. We get a ratio of 12 showing white, 3 showing dark red, and 1 showing light red. This is another of the color phenotype that we could have. So what's going on? Well, 12 to 3 to 1, it's a modification of 9 to 3 to 3 to 1. We can figure it out from that. So we're going to assume 2 genes. If we write out all the possible genotypes that we could have when we do that double heterozygote cross, you see, all right, the 9, we'll say that that one is going to be white, and we'll say that one of the 3s is going to be white. That gives us our 12, and then a 3 and a 1 for the dark red and the light red. So we could put them in here, and we're looking at it, and we're seeing, okay, you are white. In other words, it doesn't matter what you have for D, big D or little d, if you have at least one capital W, one dominant allele for that second gene here, then you're going to be white. So in those cases, once there's a big W, forget about the other, you're going to be white. But if you don't have at least one dominant allele for that epistatic gene, you've got 2 little W's, now we go to the next one and we see, all right, for the D, what do we show? Well, if you have at least one dominant D, you're going to be dark red. If you have homozygous recessive for the D, you're going to be light red. So now it's like we get to look at a standard Mendelian thing because we're not influenced by the epistatic gene. So if you see a ratio of 12 to 3 to 1 when you have crossed a double heterozygote, We call it dominant epistasis. And when one of the genes is heterozygous or homozygous dominant, it masks the effect of the other one. So we can see that all this works out, the numbers and the logic and the predictions, but can we ground it in some sort of biochemical reality? And a hypothesis that seems to reflect what's really going on here is something like this. Imagine that you've got a two-step biochemical pathway. You start out with some compound that gets modified into a pigment precursor, a chemical that now can be turned into a pigment, blue or something else. Then you have another step where that pigment precursor gets modified by the color gene into one color or another. So we go down the two steps here. Two genes are influencing it because they code for an enzyme that catalyzes the reaction from step 1 to 2 or step 2 to 3. In the case of recessive epistasis, what happens is that if you've got two recessive alleles for that epistatic gene, then it means you're going to be producing the pigment gene that doesn't function right. The enzyme is not going to catalyze the conversion of that first compound into the pigment precursor. And so what happens then? Well, then it doesn't matter what's going on here because you never get the middle step. You never get pigment precursor. You can't turn it into anything. So you are stuck with no pigment. You're just blocked. We've put up a roadblock because the epistatic pigment gene doesn't make the functional enzyme, and you end up with no pigment regardless of the second one. In the case of dominant epistasis, still talking about the same pathway. Two genes, the epistatic gene, sometimes we'll say it's upstream, and then the color gene. You have an initial compound, it's going to get modified into something that can function as a pigment precursor, and then that's going to get tweaked one way or another to produce one color or another. So with dominant epistasis, it's the same thing, but it just says for that epistatic gene, if you have homozygous dominant or heterozygous, that one dominant allele messes it up. We get stuck at the no pigment phase because we cannot modify it into pigment precursor, and you end up with no pigment, so hence the white flowers, and it doesn't matter. You never get light red or dark red in that case. So our take-home message here is that epistasis is a phenomenon where expression of one gene, in other words, you've got genetic information about how to build a phenotype, expression of that gene depends or hinges or is influenced by this other gene. So the epistatic gene, which we think of as being upstream biochemically, it can block expression for all of the alleles for the other downstream structural gene. There we have it. That is inheritance for a situation where you have two genes influencing the trait, and it is epistasis.